My name is Tom Crenshaw. I'm a faculty member in the Animal Science Department in the swine nutrition area. And one of the things I've enjoyed over my career at UW-Madison is actually the opportunity to work with undergraduate students in research programs. I'm certainly involved with teaching and research, but working with students outside of the classroom and bringing them into particular experiments, giving them exposure to things that they might do that they might not normally encounter in a classroom is one of the things I really enjoyed. I'm in front of a poster here that goes back a few years, but this is actually an illustration of several of the opportunities that we've had over the years to work with undergrads in research. And this started with one of the senior level classes that I teach, Animal Science 415, in which students are taught how to formulate diets for pigs and dogs. We get into both of those in diet formulation. But in this experiment, we noticed at the end of the experiment that, and this was the first time that this had showed up in our swine research herd, that we had pigs that developed this humpback condition, or kyphosis, which is an abnormal outward curvature of the spine. That was quite unusual, so we did experiments to understand why. In about 5 experiments in a year, it took us before we really pinned down what caused this in our particular situation. And that was due to an accidental deletion of vitamin D during the maternal or during the gestation period in which we were feeding that diet to the sows that we did not know they were being fed a deficient diet. And the reason, that's kind of the background of the experiment, but really what I want to focus on would be some of the students that were involved with that. I mentioned a year and 5 experiments. Well, we had several undergrad students. One is the senior author on this poster that's presented here, Laura Rohrbeck, name at the time, Laura Amerson, worked with me on this project. We actually went back to prove under controlled conditions that we could reproduce the problem. One of the things that Laura and a number of these other students that worked during this project and earlier projects were able to take advantage of a program we have in the department that's called the Cargill Benevinga Undergrad Research Opportunity. When one of my colleagues, Benevinga, retired in 2001, Cargill Neutrino actually donated money to the UW Foundation in his honor. And what Ben wanted to do was actually to set up an opportunity for undergrad research, undergraduate students to gain research opportunities and gain exposure to research. Ben spent all of his career working in the research area, quite an internationally known, recognized researcher in our department. But he set up this foundation where students could apply for a research grant and gain that. So I've had a number of students over the years on multiple projects that worked in that area. So another example of that is, as we begin to follow through this project, understand it more, we use an instrument called a Dexa Machine that measures the whole body bone mineral content. We've had students that work on different aspects of understanding the accuracy of Dexa and how that works and how it could particularly be applied to pigs. But we use the Dexa Machine with another undergrad student that's shown here, that we could do an image of the pig, collect an image of the whole pig, and actually measure the spinal curvature. So we used that instrument, developed a technique, and Lauren Vanderwerth was an undergrad student that worked on that, and this part of the poster was actually part of her undergrad research project that she worked with. We're continuing to work on this particular area, still involving a lot of graduate students. Laura is now actually a scientist that's continuing to work with me in that area. She finished her PhD through that, and it changed a number of students the trajectory of thinking, well, they want to go to vet school, to finding out that there's maybe other things that they can do besides going to vet school in the research area or nutrition area. And several of the students that have worked with me have continued on and going into a vet school program. So this is kind of one example of where we've worked with undergraduate students to develop research. We carried this project further and might move into the lab area, and I can show some images of what we're doing currently to better understand what causes the lesions in the spinal column and what we might be able to do to correct those lesions. We work both with people in biomedical engineering with this, and also orthopedic surgery, to better understand the lesions and how the spinal column grows and develops and what we can do to prevent those lesions. Okay, as we just mentioned, this experience that we had several years ago with the vitamin D deficiency, we've been trying to work through ways that we could understand the lesions that were involved with that more. And again, several students have been involved over the years in working with us in that area. But one of the things that we've come to understand is that lesion is consistent with an lesion of osteochromosis, which is a failure of the cartilage that would occur on the joint surfaces of bone. And so we've been trying to understand that. This is certainly a long bone or a femur that would come from a pig, but if we can produce the lesion, then what can we do to help prevent or help with the healing of the lesion? And so as we've gone through, and again, using quite a few undergrad students and graduate students in our research program, we've gone to new techniques to be able to measure and quantify the lesion. Shown in this slide is actually a 3D reconstruction of a bone that has been subjected to a CT lesion. We go over to the medical school and use a clinical CT to measure the bones. And you can see the green area that's rotating here, that would represent an area of lesion within the bone. And we've actually been able to show that we can come back and identify that lesion if we open the bone up and look specifically at that. But I call attention to the student that was involved as an undergrad, Nicole Gross, actually developed this video as part of her undergrad research project when she was working in the lab with me several years after we first discovered these lesions. We're continuing to work on that, continue to work with undergrad students. We use the students to give them an opportunity to learn. Benevinga, that I mentioned earlier, used to always say that research is an opportunity to train a student. And so we've tried to take advantage of that. We have research questions, techniques that need to be worked out, and the undergrad student comes in, we give them an opportunity to train, to learn, to dig into a little bit more than they might normally get in a classroom setting. And we think that's a great opportunity. As Ben's always said, that's an opportunity for us to train students how to think, how to solve problems, and how to move to the next level of research opportunities. So that's what I've enjoyed doing over the years, is actually working with undergrad students, working with research, and trying to solve problems and identify new ways that we can assess the problems then.